So welcome everyone and thank you for joining to today's webinar, Multi-Cancer Early Detection by Means of Cell-Free DNA Methylation in Solid Tumors. My name is Gemma Garcia and I'm the EMEA Regional Marketing Lead at Twist Bioscience, and I will be the host for today. First, we are gonna start with a few housekeeping rules. All participant lines will be muted during the webinar, so please submit your questions in the Q&A box rather than in the chat. If you include your name, we can follow up with you after the session if your question isn't answered during the webinar. And at the end of the webinar, you will be redirected to a short feedback survey, and we would greatly appreciate your input. So now I want to introduce you the speaker for today. So Dr. Smith S. Lueong, I hope I pronounce it correctly, studied biochemistry at the University of Shanghai in Cameroon and completed his PhD in molecular biotechnology at the German Cancer Research Center with a DAAD fellowship. After that, he joined the International Agency for Research on Cancer to study molecular mechanisms of carcinogenesis and biomarkers. He later moved to German Cancer Consortium to focus on liquid biopsies and therapy resistance in solid tumors, where he coordinated the DKTK liquid biopsy working group and became a member of the German Liquid Biopsy Alliance. Dr. Lueong habilitated experimental oncology at the University of Duisburg-Essen and founded the Cameroon Consortium for Translational Cancer Research. I apologize because probably I haven't pronounced all the names correctly. So, now I want to explain you a little bit more what we are gonna hear today during the webinar. So he will walk us through the biological significance of cell-free DNA methylation changes in the context of early carcinogenesis and tumor classification. He will leverage the power of NGS-based strategies to profile complex epigenetic signatures in liquid biopsy samples. And with this, we will learn about the current challenges and finish with the future directions for translating methylation-based assays from the lab to the clinical practice. This is a little disclaimer, and now I want to give you the stage. You can start sharing your slides. Dr. Lueong.
Yes, thank you very much, Gemma, for this very kind introduction of yours. And welcome to everyone who is joining the webinar today. While I struggle to share my slides, as Gemma already said, we will be talking today, looking at a few of the things that we think they are very important when it comes to multi-early cancer detection using liquid biopsies. My name is Smith Lueong, and I'm working at the Division of Solid Tumor Translational Oncology, the British Institute for Experimental Therapy at the West German Cancer Center at the University Hospital in Essen. I'll start my talk with a very short conflict of interest, a few honoraria, nothing much but that. And I would like us to look at one particular image which was taken from Globocon a few days ago. The slide here shows us the incidence of cancer in the next 20 years. And in 20 years, although I don't see everybody, I guess most of us or our kids will be at the age where they are closer to cancer than they ever were in their lives. And if there are clinicians amongst us, you would agree with me that cancer management today is already very, very challenging and would be even more so in the future. So it's not a question of whether we should do something or when should we do something. The question is, what should we be doing? And in order to improve patient life quality, there are two things we can do. The one thing we can do is to work on prevention and risk factors. And based on the topic of today, you know, I would not be talking about that. And the other thing we can do is to work on early detection and cure. The reason is if you work on early detection and cure, you clearly can see here that when you detect a cancer, which is not new information here, at an early stage, patients generally have an extremely better outcome and life quality or survival rates. The more the cancer develops, the poorer is the patient outcome, and the worse it is, or the more difficult it is to manage these cancers. And so the question is, if we talk about early detection, How early is early? What is actually early? Because we all use these words early, 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 but we think early is something completely different. And now I'll use a scheme here that describes the development of cancer that we can actually see what and how early can actually be. So this is actually a very simplified diagnostic workflow for a patient who ends up with a cancer diagnosis. You have the patient with the first symptoms. Depending on who the patient is, the patient might take again a couple of months or years until they go to the general practitioner. That brings some patient delay, and then depending on who also this general practitioner will be, he takes some time, hesitates before he decides to send the patient for in-depth investigations, and you accumulate all of these delays. However, as you do the accumulation, you basically give time to cancer to actually increase in its stage. And if we look at how cancer develops, you clearly see that between the time you have the first molecular alteration to the time you have a full-blown tumor where the patient starts feeling some clinical symptoms, it takes a decade or more. And then you have all of these diagnostic delays, starting from the patient delay, which give the cancer again time to get to its advanced stage. So the question is, what is early? Is early the time when the patient already has the first symptoms, or is early, before the symptoms actually occur? And so when the patients come to the hospital, we clinicians then gear up with all the high technologies we have, and they want to bring chaos to order. And this is why it becomes challenging. And this is actually one of the reasons why it's actually challenging, because we are always lagging behind the disease. Fortunately, however, a couple of advances have been made in the last few decades in early detection. And I here share with you data on low-dose CT in lung cancer. And you can see on the left side that people who were enrolled in the screening program with low-dose CT actually had a lower mortality rate compared to those who were not. And this has improved early diagnosis, but we are not yet at our best. You can see on the right side that stage 1 cancers still account for less than 50% of diagnosis. And if we scale it down to stage 1A1, for example, in lung cancer, what we clearly would realize is that this figure would even go further down. And this is the stage where we have the most benefit. So we still have to do something in order to improve that. That's the one thing. The second thing is that low-dose CT actually has brought some improvements, which is very obvious, as you can see from the left graphs here. But it comes also with some downsides because of ionizing radiations. There was one study that came out this year, as you can see here, not to waste time here. The study actually concluded that if everything is done just as it's been done today, we will be getting 5% of additional cancers to the natural burden that we already have. And so we definitely have to do something. Although low-dose CT is improving on early diagnosis, we also have to reduce the burden of radiation-induced cancers. And so based on our in-house data in a diagnostic workup, we try to estimate what we really need to be able to improve on that. And it's really very important here, and I have to really mention that because you need biomarkers, you need non-radiological biomarkers to complement radiology to be able to keep the performance of low-dose CT, but actually improve on, the number of false negative and reduce the number of false positive, and in the same time reducing radiological exposure and unnecessary invasive procedures. And when we talk about biomarker, I bet you if you go to PubMed and you type biomarkers, you would have, more than half of PubMed has that word biomarker. But in the clinics, we have very few of them. The simple reason is that quite often, we look for the biomarker before we determine what we want to do with them. In a typical biomarker study, we need to know what we are looking for, why we are looking for it, and where we are looking for it. And that's really important. If you don't ask the right questions before and you rather just go to your data to look for something without knowing what you're looking for, you might, at the end of the day, bargain for something which is less worth what you are. So for biomarker discovery, it's really very important. What should my biomarker look like? What is it? Where should I look for? And what do I want to do with it? Because if you want a biomarker that simply tells you this is a patient and that's not a patient, irrespective of whether it's a stage 4 or a stage 1 tumor, then that's something different than if you want a biomarker that can tell you this patient would nodules or would this malignant mass in the body will develop cancer or wouldn't develop cancer. Those are two different questions and require two different approaches. And so it's very important to ask the right questions, get the right samples and the right constellations before you actually start moving into any molecular analysis. And here, for example, we wanted to see if we want to reduce the load of radiation-induced cancer by a third, what do we need? And it came out from a simulation modeling that we would need a biomarker with 96% sensitivity and at least 82% specificity. This type of biomarker can actually reduce the false positive rate significantly, but also increase the true negative rate. And if we take a metric to actually define what the biomarker performance should be, which is something that we need to do before actually engaging in true biomarker research, then you see here on the right-hand side an example. If my goal is to find a complementary biomarker to low-dose CT that will allow me, for example, to reduce the false positive rates by 75% and yet still miss less than 5% of the true cases, then I need a biomarker that has at least 75% of specificity and at least 96% of sensitivity. So we really need to define the criteria, what we really want to have. That's really important. And only then can you find a biomarker. Now, when we do this, the next thing is, where do we need to look for it? What type of biomarker should it be? And one thing that I always tell people, or one thing I generally do, is to go back to the hallmarks of cancer and to look for those hallmarks that will best answer my question. And in this case, I'm not going to turn around, the table. We went in for methylation biomarkers for obvious reasons, and I'm going to tell you more about that in a couple of minutes. So we decided we want to look for methylation biomarkers, but where do we want to look for these biomarkers? Is it in tumor tissues? Or do we want to go and get pathology material or fresh material to look for it? Do we want to look for them in plasma? Is it in blood cells? Or where do we really want to look for that? And that's the next question, where to look for your biomarker. In our case, for example, here, this one example, we also have examples in urine, saliva, but today I'm going to focus on plasma. In this study that we carried out some years ago, we want a biomarker for multi-cancer early detection. And so we thought a biomarker, despite the organ-specific methylation pattern, that will be able to, come out when all cancers are pulled together, is likely a biomarker that is good enough for multi-cancer early detection. So that's what we did. We took plasma from several different cancer entities, different stages, different constellations, and we profiled it by whole genome methylation sequencing together with healthy donors. And we did a similar thing with Buffy code because we were not very sure where we are going to find the best biomarker. So we did it in two cases. And as you can see here, we also had to decide, and this is really very important, decide what technology you use. So here we use the Ted Appleback system instead of using bisulfite sequencing. So we didn't do any bisulfite conversion because one, you need an approach that would not damage your material further. Cell-free DNA is already highly fragmented. That's the first thing. Secondly, you need an approach that can actually accept very low input. Nowadays, bisulfite conversion kits do accept, but in the time when we did this, methylation analysis with bisulfite conversion was really very challenging if you don't have enough material. And the third thing is that you need a uniform coverage, which is really very important. And when we look at all of these criteria, we found a kit from a company, and we use that kit for this analysis. And after the sequencing, you can see here, the first thing is large data is always giving you something. If you put anything in, it's going to give you something out. So it's really very important that you control what you put in and also control what you get out of it. It's very important. So here we look at the distribution of the methylated fraction in the different sample sets and in the different patient or participant groups. And you see here a very comparable distribution. And we also look at how the participants cluster. You can see on the right side that on the plasma samples, for example, you see the healthy clustering nicely close together. 1 minute, let me take my pointer. You see them here clustering close together, whereas the patient pools are clustering separated, reflecting the complexity in the setup which I showed you before. But this is in plasma. We have to keep in mind that although we are having cancer patients, the DNA you have in blood is derived from different sources. And we need to make sure that the data we are using downstream is actually reflecting what is happening in the tumor. It's at least mirroring the methylation pattern in the tumor. So what we did is, in order not to reinvent the wheel, we then decided to go to the TCGA and we intersected our sites with sites that are hypermethylated across all TCGA tumors where we have healthy and normal. And we came up with a list or a panel of 1,400 or about 1,500 sites that were upregulated or hypermethylated in patients in plasma. Data we generated by whole genome methylation sequencing and also in the TCGA. And with these sites, we move further, and I'm going to show you now an example of some of those sites. Or you can see here how those sites perform on tissue data. We classify them based on their importance, which is basically their association with a malignant disease. You can see here we took three here in red, and I will show you a little bit more on those. And you can see clearly that in the whole genome methylation sequencing, we also see that these sites are highly methylated in the patient pools compared to the control pools. And to now zoom into one of these sites and see how it performs across different cancer entities in tumor tissue, we again took the TCGA data, and you can actually appreciate here that in all the cancer entities, there is almost a very clear separation between the tumor and the healthy samples. But early detection, like I said, it's not about detecting a cancer at stage 4. It's about detecting a cancer at a stage that you can actually cure the patient or at least improve the patient's outcome significantly. So we wanted to see if some of the markers that we are looking here or looking at here actually can do this. So if you look at the three markers marked in red, which I showed you earlier, you can see how they distribute across the different stages. This is again TCGA data, and you see that there's almost no difference in the stage of the disease. Stage 1 cases as well as stage 4 cases almost had comparable methylation patterns. And because we don't want everybody to do sequencing, we also actually evaluated this as well, in a more accessible format, because when we talk about early detection, we don't want to screen the whole population by sequencing everybody. Even if you have a panel of 10 markers, sequencing will be very challenging. So if we talk about early detection, you have to make sure that the assay you're talking about can actually be implemented even in rudimentary settings. So we developed a digital PCR assay for one of these markers, particularly marker 2. And you can see here that it actually performs really good, or at least acceptable across a couple of cancers in plasma samples. And we also found other markers that were entity specific, and you can see them here, for example, for lung cancer, where we clearly could show that this marker is actually very specific, or at least very strong only in lung cancer and not in other cancers. And this is very important because when you find multi-cancer early detection, then you also need to be able to localize the tumor. Of course, we don't want to replace imaging techniques, but like I said, we want to complement imaging technologies and reduce exposure to radiation. So this is why we're doing this here. And so it's really important that from the very beginning, you should define, your targets, your questions should be very clear. You should know exactly what you want to look for, where you want to look for, and how you want to look for it. And the next thing is, what do you want to use? What is the right sample set? And I'm going to show you here one other study that we carried. And this was thanks to a very, very professional and, high quality material we got from Twist. That's actually today hosting us. I'm not a worker of Twist and I don't receive honoraria from Twist, but I'm just pointing out that we actually tested quite a lot of providers to get targeted panels for methylation and compare their performance. And the Twist version was really one of the best or the best performing. So I really appreciate that. So what we did here is we went in for a cohort of screening patients, so people who were actually high-risk patients, and they were monitored across years and they developed disease. And here is a cohort of lung cancer and mesothelioma cases. I'm going to show you also another study on different entities, but we start with lung cancer and mesothelioma here. So here on this graph, on the left is lung cancer, and on the right is mesothelioma. Each line on these plots shows a patient, and each symbol is a time point. And please take note of the time points here. We have the time point 0, which is anytime after diagnosis, so beyond 1. And from 0 to 12 months is T1. So you have T2, which is between 12 and 18 months, T3 between 18 and 24, and T4 is actually samples collected 24 months and above before diagnosis. And you realize already with me here that some of the or most of the patients with lung cancer never had samples at diagnosis, but some of them actually had samples at diagnosis as well as in the mesothelioma. And in these type of studies, just as I said before, it's really very important that you take the right controls and that you make sure that you have the right data set. So if you look here, there's a summary of what we had. We actually paired our controls compared to the patient. So we had 2-to-1 pairing, and we took care that the controls also were smokers, as these are well-known risk factors for lung cancer. And now if you look at the T stage, you see that some of the patients were actually detected as stage 4. So if we had taken the sample just at diagnosis, then we will be looking at stage 4 patients. And now the question then is, which sample should we take for that? But before we go to that, I also want to share with you one important thing. Cancer is a disease of aging. And when we look at the controls, you should also try to match them as much as possible to your patient cohort. And you can see here for the lung cancer on the left and the mesothelioma to the right, that we tried as much as we could, not only to match the smoking status, but also to match, the age of the patients, because methylation is also, age dependent. And so you want to also make sure that you are not identifying age-related factors as your biomarkers. And we applied then the enzymatic methylation sequencing together with the panel from Bioscience. And you can see here, I'm focusing on T4 here today. Like I said, some of the cases were already at stage 4 when they were detected. So T4, which is actually the time point for not the stage of the disease, be careful, please. The T4 time point here is actually beyond 24 months before diagnosis. And you already see that there were already a lot of methylation sites that were affected at this time point, both in the lung cancer cases, but also in the mesothelioma cases. And this is really very, very important. And if we look now across all the sites that we found, we use a penalized regression model just to do that, because you have a lot of features and you don't want to get into the problem of overfitting as well. So we tried as much as we could to avoid that by using penalized models. And we identified a couple of markers. And when you look at those, a combination of those markers here, for example, in one of those markers on these samples, what you see on the left is for all the samples at the different time points. You see that when it was high in the sample, it was high at all the time points, really comparatively to the controls. But one thing that was very important, like I insisted from the very beginning, is you have patients who were diagnosed at stage 4. So T1, the time point 1 of this patient might already be T1A or even higher. So what we did is we took the marker, which you see on the left side, and we looked at it in patients who were diagnosed at stage T1A or T1B. And this was now that you see how the markers perform. Even though the patients were diagnosed at T1A or T1B, the markers at T4 are still high. So which means these markers are still capable of detecting or finding these patients even 24 months before they are diagnosed with a T1A or a T1B tumor. And when we convert this molecular data into a risk score, then you can see both for lung, but also for mesothelioma, that the markers that we identified were actually capable of nicely separating. We are not 100%, but we believe that the sensitivity and the specificity that we have defined to be our target could be reached with this performance. So at this point, we were a little bit happy. But biomarker is not a single lab or a single cohort studies. You need a lot of patients. You need a lot of different cohorts to actually falsify your data as much as possible to see if it still holds true. So what we did was then to test this in another cohort of patients. This is lung cancer again and mesothelioma, and even in this cohort, we still were able, to figure out or to separate the patients from the controls in a reasonable manner. So our biomarkers did not only work in the initial discovery cohort, but also in this test cohort was working, and these two cohorts were actually analyzed separately. The next thing is that a biomarker, if it should not work in a single lab. You should be able to apply this in a different lab and it should still work. So what we did is we have a collaborator working in a diagnostic lab. And so we provided them with one of our markers and said, look at that in your samples and let's see how it performs. And a few days ago, their first results, and as you can see here, the biomarker is having a comparable performance to what we already saw. So when you define your criteria properly from the beginning and you have the proper controls, you are most likely going to get the right biomarker you're looking for. And the one thing we want to make sure is that we are not just looking at markers of inflammation. And so what we tried to do was to look at the markers that we found for lung, but also for mesothelioma to see how much overlap we find there. And what you see there clearly is that you find almost no overlap, although they are all thoracic diseases. So you are getting here some very entity-specific cases, and we've actually also analyzed cases of COPD to make sure that this is also not associated with inflammation. I'm not showing that data here, but we have the data, which is also very promising. So that's basically one thing on thoracic cancer. I will also show you again another study we are doing, or that we are almost done with in gynecological cancers, precisely in endometrial cancer. So we wanted to actually, find early markers for not mesothelioma, sorry, for endometrial cancer. The reason being that the incidence of, although the mortality is low, the incidence is increasing, and we decided to constitute a population composed of early diagnosed patients, but also patients with risk markers or risk factors, including postmenopausal bleeding, endometriosis, but also healthy controls, age match as before. And in our test sequencing, we applied the panel, which I told you, that, which was designed by Twist. We applied it to 8 endometrial cancer patients and 8 healthy controls, matched by age. And you can see here we still were able to identify clear markers that differentiated endometrial cancer from healthy controls. Therefore, we then expanded this to our cohort of about 40 or 45 patients that we have, where we have pre and post therapy samples. And you can see clearly we identified markers which were almost exclusively hypermethylated only in the patient. So you can see here, these are all patients here. Then you have patients here with endometriosis, postmenopausal bleeding, and healthy. And you see all of them are all hypomethylated, while almost all the patients here are hypermethylated. And because endometrial cancer is a disease associated with body mass index, we wanted to see if this is also not associated with body mass index. And we did check one of the two markers, and you can see here that the BMI actually does not play a major role in the methylation pattern that we are finding here. And this is really very important because because the body mass index is gradually increasing across time, and therefore, if you want to apply such a marker to a population, we want to make sure that you are not sending people for invasive or radiological exposure just because they are obese. And how do these markers perform? If you want to look at their diagnostic performance, you see here, for example, a ROC curve showing one of the markers. And when you combine the markers, you actually can improve the performance. We are not yet at 100%, like I said, but our goals have been achieved. What we actually want to achieve as a goal with the biomarker, what we are having here, we already achieve it. And you can see here, top 10 combinations of the biomarkers that we have for endometrial cancer. You have biomarkers with up to with an AUC of close to 100, So this is combinations of close to 100, and this is really compelling because then this allows you to be able to clearly, in a minimally invasive manner, detect and say whether or not somebody with postmenopausal bleeding, endometriosis, is having endometrial cancer or will develop an endometrial cancer or not. So this is actually very important for us, and that's something we are pursuing. Now, the question, I guess, for the biologists here is always, are these markers disease relevant? Do they actually do something? So what we did is to look at one of the markers or some of the markers that we have to see if the associated genes, we annotated that, and to look if the associated genes have a dysregulation. And that will be almost my last slide for this webinar today. And here you see here that we have the expression of that gene. And in all the cancers we have tested, including endometrial and lung cancer, the gene is downregulated in tumors compared to healthy. Sorry, I didn't put a legend here, but the gray boxes are the healthy and the other ones are the patients. And we have here leukemia samples, and you see the pattern is completely different. So what works in solid tumors might not necessarily work in liquid tumors, just that we have that in mind. So basically, that's what I wanted to share with you today in this webinar. The take-home messages here is that we need tumor mirroring cfDNA methylation markers to be able to get cancer agnostic and entity-specific biomarkers, and these can actually help to develop early detection biomarkers, and they should actually be mechanistically relevant. And these biomarkers, most importantly, could allow you to detect cancer several years before the diagnosis. And this is very important if we talk about multi-cancer early detection. Early detection is not detecting a cancer at stage 3, because then even with the eye or with the hands, you can actually feel that there's something going wrong. And for methylation biomarkers, it's really important to keep in mind a single biomarker will never take us there. And I'm by no means here insinuating that we will replace normal diagnostic workups with liquid biopsy biomarkers, but that we are going to improve the current management platforms and workflows with liquid biopsy biomarkers to improve the patient experience and also improve the patient life quality, but also life expectancy. With that, I want to thank you all for your attention, and I'm happy to take any questions.
Thank you so much. It was amazing. You explained so well. Really, I really enjoyed this one. Okay, so we have some questions. I want to remind everyone that please type them on the Q&A box, not on the chat box, because otherwise we cannot take them. So we have a couple already. So we can have we'll give time people to keep adding them. Okay, so how sensitive is cell-free DNA methylation for detecting very small tumors or in the stage 1 cancers?
Thank you very much for that question. That's really a very important question. And like I said already, multi-cancer early detection is about early detection indeed. And when we talk about early, we are talking about detecting the tumor earlier than radiological technologies are capable of doing it. And cell-free DNA methylation, which went for that because methylation changes are amongst the earliest event in tumor development. And so if you can actually catch those changes, then you are capable of developing a very good biomarker. So cell-free DNA methylation, like I showed you already, even for patients with stage 1A1 tumors, if you collect the patient sample 24 months before clinical diagnosis, so which is basically a nodule, which some people might even call a benign nodule, you're still able to say whether or not that person will develop a tumor later. So cell-free DNA methylation are very sensitive, but like I also showed for mesothelioma and lung cancer, they're also very specific for detecting very early tumors.
So maybe we can go with this other question because it asks about when you have benign conditions or inflammations that I
Mhm.
guess in these early stages could be confused. So how can you reduce the false positive to know when it's coming from a tumor or it's coming from these things that are
Um,
happening?
right. This is a good question. When you have a predisposing inflammatory condition, um, that could also, and that could actually lead to false diagnosis. But let me go back to one or two slides before that that I can actually show you that. Okay, my screen is moving. But if you remember uh, the last section on endometrial cancer, we actually took care of that. And not only that, but also in the lung cancer, like I said, we've actually analyzed COPD cases, and we could see, I didn't show the data here because I prepared this talk a couple of days before, and I finished the analysis this morning, so I couldn't put that here. But we have, for example, COPD and lung fibrosis cases that we've analyzed, at least 50 cases so far, and we can demonstrate that the markers that detect stage 1A1 tumors 24 months before do not show up in these cases. And like I showed you already in the endometrial cancer cases that I showed before, we took postmenopausal bleeding and endometriosis, as well as obesity, all of which are conditions that are predisposing to endometrial cancer. And you could clearly see that the markers that we found, at least in the endometrial cancer, which was the last thing I showed, was clearly very distinct only for cancer patients and not for this pre-malignant or predisposing conditions. So this is why I said you really need to, it's all about where to look for and what to look for and what are the samples you go for. If you just take patients and then you look for that marker that flies up in all the patients, and before you test it in the predisposing conditions, you're likely going to end up with a marker. I have my colleagues or collaborators in one institute in Bochum. They actually told me, we've tried methylation markers, and each time they work well in the tumor, when we put them on COPD, it also shined very brightly. And I said, yes, because you didn't screen, you didn't ask the right questions. And now they're actually using some of the markers we are developing. They are actually developing them because they're actually doing that in an institute that is working with occupational exposure to actually screen people. And some of the markers we are developing, they're actually further trying to develop them and get them approved to use them in their setting. So, you have to ask the right questions from the very beginning. That's the key. And you have to have the right sample sets to be able to answer those questions.
Okay. I want to remind everyone that if you plug questions, please put your names because we are having a lot of questions and I don't think we have time to take them all. And some of the people is just typing with anonymous, so it will be a pity that your answer doesn't get taken right now. So a little bit on these lines. There is by the way, everybody is like thanking you for the great talk, which I also agree.
Thank you
Um,
very much.
so now you were talking about the different types of cancers, and there is someone who is asking about the pancreatic cancer, where
Yes.
early detection is so difficult. So this person is asking if there is because there is not cell-free DNA, or if maybe you have tests in this type of cancers, if you know that.
Yes, that's a good question. And just to say that I'm working in a pancreatic cancer lab. And we actually have a cohort of 350 cases. We are actually screening. The first results really look very, very promising. The problem with pancreatic cancer, we tried in the past because in pancreatic cancer, as you may know, for those who are in the field, you have more than 80 to 90% of cases with KRAS mutations. And we tried to score KRAS mutations because we thought it was going to be an easy catch, but unfortunately, it was not that easy. And in pancreatic cancer, but also in prostate cancer, even with methylation, where you have markers. I mean, for each entity, I get like 3 to 4,000 sites that are really very strong. But for prostate and pancreatic cancer, you get less than 50. So you really have, I don't, we believe that it has to do with the level of proteases that you have in pancreatic cancer, nucleases, sorry. For those who are not aware, in cancer cases, you always have nucleus degradation, but in pancreatic cancer particularly, you have much more nucleases in pancreatic cancer than many other cancers. So we think that is one of the reasons why it's really challenging to find markers for pancreatic cancer. But fortunately enough, we found a couple of markers, four for that matter, who that are really promising. And like I said, we have a cohort of about 350 cases in not really stage one disease per se, but stage one, stage two cases. And we also have a cohort of pancreatitis that we are going to look at. So yeah, pancreatic cancer is challenging. One thing I believe is actually the release of tumor-derived material. First, we know that pancreatic cancer has a very low tumor cell burden. So the percentage of tumor cells in pancreatic tumors are very low, 10 to 20%. First thing. Second thing, you have a high desmoplasmic stroma, which might also limit the release of cell-free DNA. So there are a lot of things that could account for that. But like I said, again, just repeating myself, we were able to find a few markers, and we are hoping that these ones will actually find their way forward. Thank you.
Okay. Then a little bit related to this one. I'm just trying to, we have a lot of questions, so I have a lot of options to pick. So we're trying to create this flow. So then there is someone who is asking about the size of these markers. You just commented on that, that there is, you're trying to analyze some genes for some particular cancers. So the question is like in general, actually, but it's like full genes length or partial genes length. So what your yeah, what is the size?
Um,
Usually, if you have like a usual size.
so Yeah, so basically, that's one thing I forgot to say during the talk. When you're the panel that we got designed here was actually pre-analyzed bioinformatically to make sure that we did some sequencing of different cancer entities to figure out amongst the differentially methylated sites we found in our whole genome sequencing, we actually screened them to look for sites where you had the least degradation. So you had very long fragments. That's the first thing. The second thing is we are not looking at cell-free DNA. You never will have full genes in cell-free DNA. Cell-free DNA is always fragmented. So you always have very short fragments. But we do captures with 120 base pair probes. And we have markers, we do markers of three to four markers. Basically, we limit to three because if you want to do digital PCR, you literally cannot go beyond four markers because most of the machines on the market have 6 colors. And since you need a methylated and an unmethylated probe, you always can multiplex three. So we try to create panels of three combinations. And when you do these permutations, you easily ramp up the number of markers, even if you have just 20 or 30 markers per entity. So to answer that question, you cannot have full genes in cell-free DNA because DNA is fragmented. But yeah, we try to get the longest fragments. We try to target the longest fragments in our samples by actually optimizing the panel that we are using.
So which would be the lowest you have managed to detect, the lowest fraction? This is also a question. Which would be
Um,
the lowest fraction you would have managed to do?
Fragment size, you mean?
Yeah, I think this person is referring to that. Yeah. What is the
Okay,
lowest ctDNA fraction for detection?
so if it's fragment size, I just hope that I'm answering the right question. So if you're asking about the fragment size that we've been able to detect, so basically we select our libraries to have fragments between 100 and 200 base pairs because otherwise the sequencing also doesn't make a lot of sense at the end of the day with the adapters. So we select our libraries, we do SPRI selection to have a fragment length of about 100 200 base pairs at the maximum.
Perfect. We are gonna take a couple of more questions. Otherwise we can be like all day afternoon here. So about collecting the samples, how invasive is sample collection for early stage cancers? Which samples do you collect exactly and from where?
So right now, our working platform is established on plasma samples, serum samples, and to a lesser extent, saliva and urine. We've used saliva very rarely, I think in one or two, one entity that's in head and neck cancers, we tried that. Urine in cervical cancer, we tried that. But our workhorse is plasma and serum. So collecting a plasma sample, anybody can do this, and it's not that invasive. I mean, it's not like we are getting biopsies. We are not using biopsies. We are using liquid biopsies. We are using plasma and serum. So invasive, you go to the GP, you get blood collection. So it's nothing much more than just getting to a GP and getting some health examination.
Okay. Okay, good. So we are gonna take this question. How do you eliminate the effects of lead time bias in your analysis?
That's a good question. Eliminating the effect of lead time bias. So basically, like I showed you in my previous or in the lung cancer slide, we actually evaluate different time points, right? We didn't just evaluate one time point. We have evaluate several time points to be able to determine which time point allows us to detect the cancer early enough, but not at a point where you are not capable of really helping the patient. So we basically started our work by defining the right time points in analyzing several sampling time points from our cohorts. And that's just basically our strategy to deal with lead time in these analysis.
Okay. We are gonna take one more, maybe the one where we don't have the name. So the others we can take them by mail. So great talk again, this person is saying. So it says many people have difficulty in isolating cell-free DNA from healthy samples compared to cancer samples. Do you have an estimate on how much starting material you isolated or needed, and how much cell-free DNA was needed to do different experiments? So depending if you are doing EM-seq or digital PCR, and if this cell-free DNA changes mirrored in gDNA, so in genomic DNA.
That's an excellent question. And I have to say that question is entity specific. So
Mm,
if you take, for example, breast cancer, when we work with even stage 4 breast cancer, you literally need 4 mils of blood plasma to get sufficient cell-free DNA. I'm talking about 2 to 3 nanograms per microliter in a 50 microliter eluate, for example. If we talk about lung cancer, for example, it's another world. That's the one thing. The second thing is that people use different kits for cell-free DNA extraction. People just go to literature and read. But I want to say that we started our work, that was 8 years ago. The first thing we did was we ordered 10 different cell-free DNA extraction kits, which we tested and we optimized them. So the protocols we use for cell-free DNA extraction, be it in healthy donors or in cancer patients, they are hugely optimized, not just what they provide us. So all our protocols, even the protocol of hybridization and everything from Twist, we have optimized it to suit our applications. I have to say that here. So true, you have less cell-free DNA from healthy donors, but you need optimized kits and optimized protocols. We literally use not more than 2 mils of plasma from healthy donors, and we have more than enough. We generally target to have at least 10 nanograms of DNA for an assay. The EMC kit even, but we never use less than 10 because we need to have enough genomic equivalence given the amount of material you lose during library preparation.
okay, perfect. So we have some more questions. I think if I can ask you if you want, or we can.
Yeah, you just go ahead. It's fine. I mean, they took their time to attend the webinar, so we can answer a couple more.
Okay.
Maybe not 100 questions, but a couple more we can address them. Yeah.
Okay, because I see the people is still around, so no one has dropped, so it's clearly very interesting for everyone. So I don't want to take too much time from you, but I see that there's
It's
plenty of
fine.
interest.
It's fine. Let's go ahead.
Perfect. And this one I'm actually curious because could tumor organoids be used for early detection?
Well, no. The answer is no. Tumor organoids are developed from tumor cells, which means that the tumor is already at a level that it can be biopsied, that it can already be seen by a by an imaging technology. So basically, I would say no. As soon as the tumor is beyond the diagnostic time point, we are not talking about early diagnosis. Let's keep this in mind. Quite often we are happy when we treat patients and we see, the treatment group or this group had a better survivor. They survived 10, 20 months. Just imagine somebody tells you today your lifespan is limited to the next 20, 30 months. How do you feel about that? At times I go to the clinic to sit near my colleagues, the clinicians, to actually see the patients and see their sufferings and to understand that. And that's a key driver. And if you have an organoid, you either get it from an ascites collection, a pleural collection, or from the tumor itself, which means a biopsy can already be taken from it. So this is already a little bit late.
Mm-hmm.
So I would not go for that if I really want to do early detection. Of course, you can use it to identify for exploratory identification of markers that you could do. But if you really want to do diagnosis, I would go into patient samples. You don't train for a mountain race on a flat land. You train at least on a hill. So I would go directly to the patient samples or something comparable.
Mm-hmm. Okay. Okay. Yeah, makes sense. Okay. So then last question, and this is a follow-up of another question. Yeah, well, we are keeping having questions. So do you target certain number of CGPGs? So methylation island, CPGs.
Yes, so I would say, like I said, the design of our panel was clearly optimized. So we did the whole genome sequencing, and then we tried to do also some fragmentomic analysis to see which regions actually had good fragments and where we also have potential or compelling biomarkers. And then we designed our probes targeting that area. So we did not precisely say we only want CPGs. We went with the data, we actually work with the data. We let the data to guide us. So the data said, this is where I find the most differences. This is where I find the most interesting sites. And so we went to those sites and identified areas where we can actually have probes, where across a certain number of patients, we had longer fragments. So we believe those regions are highly, you still can have them in a good form in most patients. So that's how we did it. So we do not really focus. In some, for example, the endometrial cancer sites that are actually presented here, these are promoter sites of a given gene. We are actually filing this for a patent, so I can't tell you what that is. But these are two genes that are part of a complex. And these markers here actually, several methylation sites are the promoter of a gene. For some, we have, genes that are clustered together within, sites that are clustered together within one gene. And for others, there are sites dispersed across different genes. So it's always, data-driven investigations, not predefined. Like I said, we do not define what we want. We let the data to tell us where we need to be going. But before you do that, you might have to make sure you have the right data. If you put shit in, you get shit out. So it's really very important with big data to define the right questions.
Mhm. Okay, perfect. Yeah, you have said it several times today that you have to make the right questions, and the data you put is whatever
Yeah.
you will get from it. So, okay, perfect. Now we are gonna leave it because otherwise we're gonna be here like all the afternoon. That wouldn't be fair for you. So thank you so much, thank you so much. It has been really, really good. I really enjoyed this webinar, and thank you everyone for joining and staying so long and making so many questions. So just a quick reminder for everyone, you will be redirected to a short survey, and if you could give us some feedback, that would be amazing to know what we are doing good. And on behalf of Swiss Bioscience, we want to thank you for joining, and we look forward to everyone to join the next webinar. So thank you so much, Dr. Lueong. It has been amazing.
Thank you very much. And thanks to everybody for the time. And I hope that I could stir, enthusiasm in some people to engage in this as well. And I'm also open to discussions. Thank you very much.
Thank you so much. Have a nice day.
Have a nice day. Bye bye to everyone.